Hey, what's up, YouTube? Thanks for tuning in to Hognose 1. Today, we're going to have another morph topic. We're going to get into genetics a little bit. So anything you hear on this one may supersede some of the other genetic videos I've had in the past. We should just grow in our performance. We should grow in our understanding as we move forward. So hopefully there's things here you can glean from and learn that may differ from what we've been taught in the past. Lots and lots of breeding trials have taken place here at Hognose 1. I've heard lots of other situations that have happened and occurred too. And I would like to look at something that we see dealing with genetics in the herpetoculture hobby. Sometimes the things we hear have valid scientific reason behind them, and they can be understood even if they don't align with academia. And I believe that we should not try to be academia because there can be some beneficial crossover on a layman's term, basically where we can contribute to our understanding of something. But I think it's good to get behind certain terms, especially dealing with patterns of inheritance. So in high school, we learned basically that there were two types of inheritance, recessive and dominant, and those helped us with Punnett squares and the mathematical type equations to understand basic body inheritance function, skin color, eye color, widow's peak, whatever you have like that. But I think that it comes of benefit and interest in the hognose hobby and ball pythons and other ones too like that, because there are so many different forms of expression. So in this particular series, I do want to talk about different types of expression. I want to talk about the difference between a dominant inheritance and a dominant expression, because you can have both at the same time. There are many different genes that rely on each other for expression. Real quick before we get into that, dominant inheritance describes the relationship between genes in a given gene pool. So dominant expression talks about the relationship between only one gene necessary to present itself and to express the given phenotypic trait. So these are intertwined, but they're not exactly the same. It's very curious that there are nuanced terms, polygenes, polygenics. Everybody loves saying it. We love the idea of this because it's a fairly nuanced. It's only been around for 3 or 4 years that we've actually attributed genes into that category. But to understand polygenes, it's a very, very difficult situation. When we're able to send off a shed for some ideas from a lab, they can compare it against the mapped hognose genome. It will make it amazing to see some of the relationships and some of the things, because a computer can do a lot more than we can with our test pairings. If you want to get anywhere and determine whether an expressed trait is recessive or dominant, you have to do test pairings. You have to do crosses, test crosses. We call those breeder-selected animals and pairings. So those take a long time. Sometimes it's 2 years, sometimes it's 4 years, sometimes it's longer than that, because we have to get babies first generation, cross them back to one another to see if we can get to this desired outcome, if it's recessive. If it's not and it's dominant, we'll see it first generation, which is something kind of cool. But polygenes was a nuanced term. It was something that came around much like the term codom. The first time we heard that in hognose snakes was back with anacondas somewhere around 2008. And codom was something that kind of made sense in the reptile community, but it was not accurate. And where the accuracy lies is that when you've got a true codominant, means that in that expression, two different dominant traits expressed at the same time. Whereas as we know now, that the anaconda and superconda complex is an incomplete dominant complex, meaning that you need two expressions of the same trait to express the super or homozygous form, just like you would with recessive, but the difference here being is that the heterozygous form is visual, and with a recessive trait, all your hets just look like a wild type or standard. They don't express even partially like an incomplete dominant trait does. So quickly before we wrap up this session, Of interest, and where I want to go with the next few sections, is for a long time, the dominant expression, meaning the expression that was most consistent, or one of the most consistent ones, was a solid black belly with white walls. Almost every single example of the anaconda gene had that specific dominant expression. It was consistent when there were test pairings and people were crossing things in until a point. There was one point where the albino gene was crossed in, and then all of a sudden we started seeing another gene come up. So this interplay of genes, you might even call it polygenes, came up and caused that dominant expression that we had seen for several years in that trait, anaconda or even superconda, caused the white walls and the dark belly to begin to have mottling. We saw stripes down the middle. All of a sudden, there was something new completely playing in here. So this is the relationship, and this is where we would call it polygenes, because there's multiple genes that express. Some of them express in a codominant fashion, meaning the anaconda gene was your typical black with white sidewalls. Now this albino gene comes in, and it throws yellows on the belly, which threw everybody off at the same time. So they do act in a codominant manner. So let's get into it with our next vid, talk more about this stuff. Very cool stuff in learning the relationship between genes. As always, guys, thanks for the like and subscribe. I hope you find this information very interesting, and we will catch you on the flip side.